Friday? Anybody here unhappy that it's Friday? Everybody's happy that it's Friday. Wild plans for the weekend? No? Studying biochemistry? That's pretty wild. So we're actually pretty well caught up now, so I feel pretty good about that. Hopefully you're not exhausted in getting to this point. One last thing I did not mention about prostaglandins, which is a term that you'll frequently hear associated with them, and that's the term eicosanoid. Okay? The prostaglandins are called eicosanoids, and that relates to the fact that they have 20 carbons. Eicosa referring to 20. So they're eicosanoid compounds. And that's really important when we think about these because prostaglandins, the eicosanoids creating pain, they're the ones to blame when you get inflamed and ouch, they hurt inside your brain. Prostaglandins, every throb and ache gets magnified. If you hope to win cyclooxygens, generations got to be denied. The Vioxx has all been recalled. So go get yourself Tylenol. And if you ache, blame PGH synthase. We must complain that you make the aches. Prostaglandins, prostaglandin, D2F1G2, prostaglandin. It's you. All right. All right. No. That was really bad. I know you guys are being very gratuitous in that. But I thought a little surprise to wake everybody up on Friday might be good. Hopefully you're awake. If you're asleep after that, I don't know what to say. So maybe you're dead. Okay. Well, we move now to something that maybe make you very happy or very sad. I don't know. But it's the last of our topics of metabolism. So we've been going on metabolic pathways for a while, since late last term. And now we turn our attention to nucleotide metabolism. And in many respects, nucleotide metabolism is one of the most interesting pathways. We don't go into it in the detail that we go to in the other pathways, although it may seem like we do, because there's a lot of complexity here. I'm going to go over some reactions very quickly, and you may rest assured that's how much attention I think you should pay to some of the reactions I'll be giving you. So you're going to see a lot of reactions. I'm not going to hold you responsible for structures. I want you getting a big picture here. And probably one of the most important things we can get out of nucleotide metabolism is an appreciation of the value of regulation and how and why cells are doing that regulation. The why is actually pretty straightforward. The why of regulation in nucleotide metabolism is that cells have to balance the appropriate number of each nucleotide inside of them. We know all kinds of examples of cells that don't balance those things properly, and every time those cells are much more prone to mutation. Every time, okay? So if cells don't balance the relative amounts of nucleotides properly, they are much more prone to having mutation. That's just given, all right? So the value of regulation, and we're going to see some interesting, they're not overly complicated, although they're a little bit more complex than we've seen in other things. We'll see, interestingly, how different regulatory systems work together to ensure that cells have the proper balance of things that they need. Okay? Now, nucleotide biosynthesis is also interesting in another perspective, and that other perspective is there's really two ways of getting there. All right? Two ways of getting there. One way of getting there is what's called salvage. And salvage, of course, happens all the time. We eat food. The food is full of fats and protein and nucleic acids. And so our digestive system, just like it takes apart proteins to individual amino acids, and it breaks down fat into fatty acids that we use for other purposes, so too does our digestive system take apart DNA and RNA. And further, it takes apart the nucleotides. The nucleotides have three components. Okay? So the nucleotides are the building blocks, ATP, GTP, CTP. By taking apart those three components, the cell can mix and match and put them back together, having a material that's already partly made. The things that they mostly mix and match are the bases. So if I have an ATP and I start chopping off the phosphates and I cut off the ribose away from the adenosine, I can use that ribose for something else. I can use that adenosine to make new ATP if I want. Okay? So that phenomenon is called salvage. All right? So salvage is basically using some preformed pieces to make finished nucleotides. We haven't really talked about salvage with respect to other pathways, but salvage is a very important pathway in nucleotide metabolism. I will talk a little bit about salvage. Okay? Most of what I will talk about with respect to salvage will come later when I talk about mutations in enzymes that are important in salvage and some of the bizarre phenotypes that they give in people who possess those mutant enzymes. Okay? The other general mechanism for making nucleotides is what's called de novo. And de novo means literally from anew. All right? So the de novo pathway we can think of as starting from scratch. We start with the very simplest of materials and we build them into the very same end products that the salvage pathway does, okay? But we're starting at a different point. So if we look at what we start with in a salvage pathway, we start with something called activated ribose, which you'll more commonly call PRPP. It's got a longer name that I'll show you, but for our purposes, you'll probably call it PRPP. I can take PRPP and I can join it to a base and make a nucleotide. All right? Bang, one step, I have made a nucleotide. When I go through the de novo pathway, which we're going to spend a little bit more time on, it's about 10 steps, starting from scratch to get to a finished nucleotide. All right? Now, well, why do cells do this versus this? If they've got salvage, they'll use it, because salvage saves a lot of energy. If they don't have enough things to salvage, they will start from scratch. Okay. Now, the scratch materials are basic, simple things that are found in every cell. Amino acids, ATP, carbon dioxide, and a few other things. All right? Very simple starting materials to start making nucleotides from scratch. That's very cool. Well, let's go through a little bit of nomenclature. All right? So it's important to understand, if we're going to walk the walk, we have to talk the talk. I said there's three components to a nucleotide. All right? A nucleotide contains a base. That base would be either adenosine. Okay? I'm sorry, the base would be either guanine, adenine, thymine, cytosine, or uracil. So those are bases. They're not nucleotides. They're the things that are in nucleotides that we think of, oh, A goes with T and G goes with C, but they're only a part of a nucleotide. If I take a base and I link it to a sugar, I create a nucleoside. A nucleoside can be a ribonucleoside if I link it to ribose. It becomes a deoxyribonucleoside if I link it to deoxyribose. Okay? Adenosine is what results when I connect an adenine to a ribose. Deoxyadenosine is what arises when I connect an adenine to a deoxyribose. Okay? A nucleotide can exist in two forms, either a ribonucleotide if it started out as a ribose, or a deoxyribonucleotide if it contains a deoxyribose. Well, what differentiates a ribonucleoside from a ribonucleotide? It's at least one phosphate. So if I take a ribonucleoside and I put a phosphate onto it, I've got a ribonucleotide. If I take a deoxyribonucleoside and I put a phosphate on it, I've got a deoxyribonucleotide. Notice I said at least one phosphate. It could have two, it could have three. Okay? But that's what's happening in, or that's what the important nomenclature is with respect to what we're talking about here. Okay? So a base, a nucleoside, and a nucleotide. Yes, question, Connie.
Does that mean that if it has like two phosphates, it's still considered a nucleotide?
Yes, if it has two phosphates, it's a nucleotide, yes. If I say nucleotide, it basically encompasses both ribonucleotides and deoxyribonucleotides. If I say nucleoside, it encompasses both of these, obviously, as well. Okay? But yeah, ADP is a nucleotide, yeah.
So nomenclature-wise, would it still be considered, for example, adenylate if it was ADP or ATP, or is that
Yes.
change?
Okay, so the question is, would this be considered adenylate if you had that? See, I don't like these nomenclatures because they really refer to monophosphates, and I prefer to call it AMP or ADP or ATP. So I'm not going to hold you responsible for knowing those names right there. I just think that's a little busy. And we don't use that nomenclature for an ADP, for example. Yeah. This refers to adenylic acid, for what that's worth. Yeah. Okay. But we should know what the difference is between a base, a nucleoside, a nucleotide, those in terms of just general components. Okay. Structures, all right. The sugars that are found, you've already seen ribose before. We talked about carbohydrates last term. Ribose is a 5-carbon sugar. 5-carbon sugar that looks like this guy over here. You can ignore the 6, and that's a 6-membered ring. It's not a 6-carbon sugar. Remember, we have 5 carbons, so in making the 6th, this oxygen is combining with this carbon here. We're not going to worry about that. We will worry about this. This guy is the form in which we find ribose in ribonucleotides. Okay? The only difference between ribose and deoxyribose is at carbon number 2. Carbon number 2 is where deoxyribose is lacking in oxygen. Okay? So we'll take a look at that. Deoxyribose is right here. Okay? And it's lacking in oxygen. There's actually a hydrogen they're not showing there, which is kind of misleading, but this guy's lacking in oxygen there. Now, it turns out that the lack of that oxygen has some fairly significant effects on the chemistry of ribonucleotides versus deoxyribonucleotides. Of course, RNA contains ribonucleotides. DNA contains deoxyribonucleotides because that's where the D comes from. Okay. The significant difference, there are two that come to my mind. One is if we put an OH on this guy, making it a ribose, it makes this guy actually chemically less stable than DNA. Chemically less stable. What does that mean? It means that if I take RNA and I treat it with sodium hydroxide, that extra OH there will allow this bond to break very readily. In fact, it's one of the ways in the laboratory in which we break RNA is by treating it with alkali. So when we think about evolutionary time and people go out and they isolate DNA from some species that was in an extinct muskox from 40,000 years ago, all right, what they're getting is DNA. RNA is not chemically stable under that long period of time and under more severe chemical conditions. So that extra OH makes RNA a less chemically stable molecule. Another thing that that extra OH group does is it changes, or I shouldn't say changes, but it strongly influences the structure of the nucleic acid in the duplex. When we talk about DNA, we talked about it last term briefly. We said the most common form of DNA is what we call the B form. That's what Watson and Crick discovered along by stealing the data of Rosalind Franklin. Okay? All right? RNA can also exist in a duplex. You've got the same bases. You've got the same arrangements. You can make double-stranded RNA if you make it complementary. And there are some viruses, in fact, that use double-stranded RNA in as their genetic material. Okay? But they don't have the same arrangement of those structures. The RNA form is what's called the A form. Okay? And DNA can form an A form too, but it really prefers the B form. So RNA cannot exist in the B form, and the reason it can't is that extra oxygen actually precludes RNA making it into the B form. So, two things that happen as a result of that hydroxyl group. Okay, let's see. What else do I want to say? There are the bases. I'm not going to ask you to memorize the structures, but I think that you should know the generalities of the structure. All right? we break the bases into two groups, pyrimidines and purines. There are three pyrimidines, uracil, cytosine, and thymine. And the common feature that they have structurally is that they have a single six-membered ring. They're smaller. Okay? The purines, which include adenine and guanine, look different. They have a six-membered ring joined to a five-membered ring. They're bigger. Now, if you think about it, when we have the structure of a DNA duplex, or for that matter, an RNA duplex, A always pairs with T. G pairs with C. That means a purine is always paired with a pyrimidine. A big goes with a little, and as a consequence, the dimensions are virtually identical, whether we have an AT or a GC. That is, the width of the duplex doesn't change based on whether it's an AT base pair or a GC base pair. That's good. We don't want a lot of fluctuation there. Okay. There are the deoxyribonucleotides, and again, you see the structure here. I wish they would put an H on there to indicate there's an H, but suffice it to say that's what DCTP would look like. Okay, well, let's dive in and say a few words about synthesis and about how we make these. So what I'm going to be talking about in the lecture today will completely be de novo, starting from scratch. Okay? So we're completely starting from scratch. The pathway that leads to pyrimidines is completely separate from the pathway that leads to purines. And they also have different starting points. Okay? Different starting points. We will see that in working with pyrimidines, that the bases are made separate from the ribose. All right? We'll see in the case of the purines that the bases are built on the ribose. So in the case of the pyrimidines, ribose over here, base built over here, then they get joined together. In the case of purines, start with ribose and start building a ring on that ribose. So it's a fundamentally different approach. Let's talk first about pyrimidines. This figure illustrates very nicely the simple materials we need to make the ring structure of pyrimidines. Bicarbonate. We've seen that 1,000 times already. Ammonia. ATP. Okay? These guys come together in red and contribute those two atoms right there. Okay? Aspartic acid. You've seen aspartic acid. There's the other guys there. All right? This makes a pyrimidine ring. We join it with PRPP, which is our source of ribose. We make a nucleotide, we add some more phosphates to it, and we get down here. So I'm going to take you through these steps, but this is a general view of what's happening. Very simple starting materials, making some fairly complicated structures. Okay. Let's look at some of these simple, these very first reactions. Okay. So I'm only going to give you the names of a couple of enzymes. You're going to see about 10 reactions in making the pyrimidines and about 10 reactions in making the purines, and going through either structures or enzymes for all those is really not very useful, I think, in terms of learning the material. So where I talk about an enzyme, you're responsible for it, but I'm not going to talk about most of the enzymes, and I'm not going to hold you responsible for structures, except, as I said, in a general way where you know that pyrimidines have the smaller ring and the purines have the larger ring. Okay, so that means a lot of this you can relax on. Here's bicarbonate. Okay? We phosphorylate it, make carboxyphosphate, and we add an amine to it, and we make carbamic acid. Okay? Now, hello? My phone just rang. Yeah? Mm-hmm. Okay, bye. I pick it up and the thing's ringing on the other end. That's, can I help you? It's like, I answer because, they do have these emergencies, so you have to make sure that probably just a crank call. All right. So one of the things about what you see on the screen is that this intermediate in particular is very unstable. Okay? So we've talked about some strategies for dealing with unstable intermediates. If you recall, we talked about perfect enzymes. Perfect enzymes were perfect because they wanted to work so fast they eliminated or reduced significantly the time it took for an unstable intermediate to be present. We saw in the case of nucleoside monophosphate kinases, they had a lid that came down and closed and kept that unstable intermediate from having access to water. I'm going to show you an enzyme here that uses a third strategy for protecting an unstable intermediate. Okay? It's this enzyme here that I will mention the name of. It's carbamoyl phosphate synthetase. And it's catalyzing those reactions that you just saw, both of those reactions. Okay? What it does is this enzyme uses a tunnel. The intermediates actually move through the inside of the enzyme in the process of those catalytic reactions. And as a result, the intermediates never get access to water. So the tunnel provides yet another way of keeping water away from, in this case, carbamic acid, and making problems where the material that the unstable intermediate will fall apart. Okay, well, that's kind of a cool mechanism. We look at our product here, carbamoyl phosphate, and we are ready to start assembling a ring, or pretty close to it. And in doing this, we bring in an aspartic acid. We see the aspartic acid coming in here, and we make carbamoyl aspartate. You guys have actually seen this before. You probably don't remember it, but when we talked about ATCase, aspartate transcarbamoylase, it catalyzed this reaction right here. And you recall ATCase was really interesting because we used it as a prime example of an enzyme under allosteric control. Okay? If you recall, ATCase had some interesting things that affected it allosterically. All right? Two that were of relevance to us were CTP and ATP. ATP, we said, turned the enzyme on, and CTP turned the enzyme off. Well, that turns out to be really relevant for our purposes because CTP is the end product of this pyrimidine synthesis pathway. It's the end product. So the end product is feeding back, turning off the enzyme by feedback inhibition, saying, hey, we've got plenty of CTP, we don't need any more. I probably said a little bit about this when I talked about it last term. ATP, on the other hand, is an indicator of energy, and importantly, it's an indicator of the level of purines. If ATP is in greater concentration than CTP, this enzyme is going to be more likely to be activated because ATP wins the race, and that's good because when we have more purines, we want to balance and make more pyrimidines. This is our first example of a balancing reaction that's very important for nucleotide biosynthesis. This enzyme, ATCase, ATCase plays a very critical role in balancing the relative amounts of pyrimidines versus purines. Very important role. Which way does it tip? Well, we see. If we have too many pyrimidines, the enzyme gets turned off. If we have too many purines, the enzyme gets turned on. Okay. Well, this figure has been drawn to show you how we are very close to making a ring structure, and there's the ring structure that you see coming close there. It simply involves loss of a water molecule catalyzed by a proton, and when we do that, the ring closes. We make something called dihydroorotate. No, I don't care if you know that name. Partly because when we get to orotate, orotate is originally named from orotic acid, and I can't make that say that without sounding funny. Okay? Orotic acid. Yeah. What are you talking about, man? All right. So, you dissing me? All right. So, I don't know what that means. All right, so we've made the ring structure, all right? This is very close to being a pyrimidine. In fact, when we add a PRPP to it, we actually call it a pyrimidine, all right? Here is PRPP, all right? Ribose 5-phosphate, which we get from, by the way, we get ribose 5-phosphate from the pentose phosphate pathway. We don't really talk much about it in this class, but the pentose phosphate pathway is the source of ribose 5-phosphate needed to make nucleotides. Ribose 5-phosphate becomes PRPP by action of this enzyme, PRPP synthetase, and yes, I think you should know the name of that enzyme. What's this enzyme doing? It's taking phosphates off of ATP, and notice it's taking 2 of the phosphates and putting those 2 phosphates over here. Notice it's putting them on in the alpha configuration, and we're going to see that that bond gets flipped when the nucleotide itself is made. PRPP is a starting material to link that ring we just made to. All right, so now we're going to take this ring and we're going to link the 2 together. Okay. There's the orotate that we made. There's the PRPP. We put the 2 together and we get an even bigger mouthful name that I can't say very well either, and I won't. All right? We're almost at a pyrimidine that we can recognize. By the way, we will call this something. All right? Instead of calling it a rotadylate, I like to call it OMP. OMP is easy to say. Notice that we went from an alpha configuration to a beta. This enzyme flipped that in the process. And we're almost at our first pyrimidine nucleotide. That first pyrimidine nucleotide is UMP. Remember, we're not going to call it uridylate. We're going to call it UMP. What does that involve? That involves loss of a carboxyl. That carboxyl right there turns out to be interesting. And you've actually seen this reaction before. And it's an enzyme I'm not going to hold you responsible for, but I'll tell you what it is and remind you where you saw it before. You saw it before. The enzyme is called Omp Decarboxylase. I don't remember that at all. Well, when I first talked about enzymes, I used this enzyme as an example of an enzyme that speeds up a reaction incredibly. The half-life of this reaction is something on the order of tens of millions of years. With the enzyme, this reaction proceeds in seconds. Okay? So this is a pretty interesting enzyme in terms of how much it speeds up a reaction, and it's simply decarboxylating this guy right here to leave us with UMP. So the first pyrimidine that we recognize as a pyrimidine, that is based on our previous experiences, is UMP. We've just made UMP. Okay. Well, UMP we can't use to make DNA or RNA because that one phosphate, we have to have triphosphates to use an RNA polymerase to make RNA or a DNA polymerase to make DNA. We have to get to the triphosphate level, and we're only at the monophosphate level. Well, now we come up with another enzyme that you've seen before. Okay? And the other enzyme that you've seen before is nucleoside monophosphate kinase. This is the one that has the lid to keep the unstable molecule from floating away. All right? What does it do? It takes a monophosphate, and it can be any monophosphate, adds ATP, energy from ATP, and look what it makes. It makes UDP, so it's transferring a phosphate from ATP onto UMP to make UDP and ADP. Now, I'm exaggerating a little bit in that I said it takes any nucleotide. It doesn't strictly do that, but there are nucleoside monophosphates for each nucleotide. So there's a UMP kinase, there's a CMP kinase, there's an AMP kinase, etc. All right? And they all do the same thing. An AMP kinase would take AMP plus ATP and make ADP plus ADP. Right? Transferring a phosphate from one to the other. So a nucleoside monophosphate kinase will take a phosphate off of ATP and put it onto a monophosphate. All right, so we've gotten to the diphosphate level. The triphosphate level is catalyzed by the formation of the triphosphate is catalyzed by a different enzyme that's actually very simple. It's called nucleoside diphosphate kinase. You're welcome to call that NDPK. Now, the reason this is simple is that this enzyme works, one enzyme works on all of the diphosphates. It works on all of the diphosphates. Moreover, it can use any of the triphosphates as a donor of the phosphate. That's what the X and the Y there refer to. So, for example, if I had UDP plus GTP, I would end up with UTP plus, and that's a typo there, that should be GDP. That should be a D right there. Sorry about that. That's my error. Okay? That should be a D. So it's just swapping a phosphate from 1,3 onto a 2, and that leaves behind a 2. Now, this enzyme is interesting because it works on all the diphosphates, whether it's a ribonucleoside or a deoxyribonucleoside. This could be, for example, DGDP plus ATP gives me DGTP plus ADP. All right? So any of the diphosphates to triphosphates are catalyzed by this enzyme, nucleoside diphosphate kinase, or NDPK. That's a great simplifying thing in this scheme. One enzyme takes all the diphosphates to triphosphates. Yes, sir?
Is there an extra inorganic phosphate that comes in because they're both triphosphates on one? So.
Well, that's what I say. That's my error. That should be a diphosphate there. Yeah, sorry. Yeah. I'll change that. Okay. Now, this enzyme actually turns out to be a balancer also. Let's imagine I've got an awful lot of GTP, but not so much of CTP. Well, I can swap with this enzyme back and forth and balance out my relative amounts of triphosphates. So NDPK helps to balance the relative amounts of triphosphates. I'll repeat that because that's an important point. NDPK helps to balance the relative amounts of triphosphates. Okay? All right, so we've now gotten to the point where if this has been a U all the way through, we now have UTP right here. UTP is good for making RNA. Bang, we're ready. Well, how about CTP? Okay, because we need CTP for RNA also. CTP turns out to be made directly from UTP. It's the only nucleotide that's made directly from a different nucleotide as a triphosphate. All right? So it's made directly from UTP, and it looks like this. Here's UTP. Okay? This is an amination. We're putting on an amine group in place of an oxygen. So here's an oxygen in UTP, swapping it for an amine in CTP. Okay? This enzyme is called CTP synthetase. CTP synthetase. This enzyme is inhibited by CTP. Okay? Well, this enzyme now helps to balance the relative amounts of UTP and CTP. We don't want to have too much of either one of those. We get too much CTP, this enzyme is inhibited. We get too much UTP, this enzyme is favored. So balance, again, is very, very important. In fact, we'll see there's balancing mechanisms for every step in these processes, ultimately. Okay. Well, I haven't said anything about TTP. We haven't talked about DNA. It turns out TTP is made by an odd mechanism that I will talk about separately on the DNA when I talk about deoxyribonucleotides. Okay? DTTP is made from UMP. But it's a more involved mechanism than we'll go into here today. We will talk about it later. And right now, we're only talking about ribonucleotides anyway. So keep that in mind. Everything we're talking about here, all are ribonucleotides that we're making. Okay, good place to stop and ask for questions. Everybody's ready for the weekend, aren't they? Oh. All right, if you guys will stick with me, we'll sing a song at the end of the period. How's that? 2 in one day. Or you can get up and leave at that point if you wish also. One of the things you see on the reviews was most people really like the songs, but there's a few people that said, no more songs. Okay, if you want to, you can leave. That's okay. I'm sorry. All right. That's pyrimidines. Let's talk about purines. All right? So keep in mind, we've just described how to make the ribonucleotides CTP and UTP. We haven't talked about deoxyribonucleotides yet, and they have other considerations, but ribonucleotides we've talked about. Okay, well, let's go through de novo synthesis for a purine. All right? Here's the same scheme we saw before. There's a little bit more complexity here. All right? And purines themselves are a bit more complex. And no, I don't expect that you're going to memorize where each atom comes out of there, because I'm not expecting you to memorize the structures. But I think it's useful to look at this and think about, again, the simplicity or the simple molecules that go into this. Aspartate. There's an amine coming from aspartic acid. There's a carbon coming from carbon dioxide. There's this yellow section. That's just glycine. Okay? N10, formyl tetrahydrofolate. That's a mouthful of a name. That's folic acid. It's a vitamin, and we'll talk about that in a bit. There is ribose down there, and then last, there's an amine coming from glutamine. So with the exception of this mouthful of a name, very simple precursors that are coming together to make this. That makes a purine ring, creates another new nucleotide we haven't heard of before called IMP. And IMP, as you will see, is a branch between the synthesis of the adenine nucleotides and the guanine nucleotides. Okay? This figure is very misleading. You do not go from ATP to DATP. Okay? Very misleading. So you can ignore the very bottom part of that figure. Okay. Now, here's some of that complexity I told you about. If you were taking a majors class in biochemistry, which I know you're all thanking yourself right now that you're not, you probably would learn most of these guys on here. All right? We're not going to do that. All right? We're only going to talk about a couple of interesting enzymes on here and look mainly at the end products, the end product here being IMP. All right? Well, let's start here. Phosphoribosylamine. Okay? For reasons that aren't clear to me, your book shows this as the starting material, and it's not. And the last edition of the book did the same thing. Okay? The starting material is actually ribose 5-phosphate. Okay? Ribose 5-phosphate. And if we take ribose 5-phosphate and we put an amine onto it, we create phosphoribosylamine. That's what this guy is here. So this is actually the second molecule. I don't know why your book does this. Why do I mention that? Well, I mention that because the enzyme that makes this molecule is a very important regulatory enzyme. Okay? The enzyme that makes phosphoribosylamine has a mouthful of a name. It's called PRPP amidotransferase. And I say that start with ribose 5-phosphate. It's not. It's starting with PRPP. PRPP amidotransferase. PRPP amidotransferase catalyzes the formation of phosphoribosylamine. Okay? PRPP amidotransferase is important because it's a regulatory enzyme, and its regulation is really interesting. You may not fully appreciate it until I go all the way through the pathway, but I'll tell you what it is. Okay? The enzyme is partly or fully inhibited by its end products of the pathway. What does that mean? Well, the end products of the pathway for making purines, at least from this perspective, is AMP and GMP. Yes, we have to put phosphates on, but for this pathway, AMP and GMP are the end products. The enzyme is fully inhibited when both AMP and GMP bind to it. So PRPP amidotransferase is fully inhibited when both AMP and GMP bind to the enzyme. Turns the enzyme off, just like that. If only one of them bind, let's say only AMP binds, the enzyme is a little bit active. If only GMP binds, the enzyme is a little bit active. Now that you're not going to appreciate until we get near the end of the pathway. But let's say, first of all, that the binding of AMP and GMP will occur under conditions when we've got a lot of AMP and GMP, right? This is turning off purine biosynthesis. It's helping to control how many purines we make. Now what we're going to see is this enzyme plays a role in balancing purines as well. Okay? But for our purposes right now, the main thing that we know is that it controls if we make purines or not, and thereby controls the level of purines that we're going to have. Okay. Well, let's see. What do we want to see here? Let's start right here. THF going with a carbon on it going to a THF here. All right. Don't worry about the structures. A couple days ago, I talked about SAM, S-Adenosyl Methionine. Does anybody remember what SAM does? It donates methyl groups. It's a source of single carbons in a metabolic process. We use it to make phosphatidylcholine, right? All right? And I said at the time that there are other molecules that donate single carbons to reactions, and this is one of them. The folates function to donate single carbons. They're not donating methyl groups, but they're donating single carbons to reactions. This carbon right here in blue has been donated. There it is right there. This carbon in blue has been donated by this THF right here. Okay? Now, I show you that not because it's a little piece of esoteric trivia, but it turns out that folate metabolism has some very important implications both for making antibiotics against bacteria and for treating cancer. Okay? Now, I'm not going to tell you that right now. That will have to come back to probably the lecture next time. But both of those are important considerations in folate metabolism. Folates are necessary to make purine nucleotides. Okay? We have to have these folates to make these purine nucleotides. Okay? All right. It turns out that folates are used in a couple places. There's one right there. There's another one over here. Okay? And no, you don't need to know where they are, and you don't need to know these names or these structures. I'd like to point out some interesting things that happen along the way, though. All right? Look at this guy over here. There's a mouthful of a name for you. And look what gets split off in the process. Fumarate. Our friend fumarate. I told you that metabolic pathways are integrated. I gave you examples about how CDP was necessary for making glycerol phospholipids. And I said it was a barometer of energy for cells. And I said GTP was used for making protein. And ATP. All of these nucleotides played roles in metabolic pathways. And now we see that the synthesis of these nucleotides contribute to metabolic pathways as well. We can't think of nucleotide metabolism as being separate from metabolic processes that occur in the cell. They're totally dependent upon them, and they totally contribute to them as well. That's really important because, again, cells have to make decisions. Do I have the resources necessary to replicate? By taking simple measures of certain molecules in cells, cells know if they have the resources they need to replicate. That's very cool. Okay. Well, the upshot of this whole process is that we get down to make this nucleotide called inosinate, which you're more than welcome to call IMP. Okay? IMP is a purine nucleotide. And by the way, I didn't point out, I should point out, all of these guys already have ribose in them. See, we started with the ribose over here. So before, when we made pyrimidines, we made the ring, and then we put it together with PRPP, and we did it. We're starting over here with PRPP and building that ring on the PRPP. That's what all these reactions are doing. Notice there's the ribose right here. Okay? So we've got the ribose, right? We've got the phosphate, because it started out as a PRPP. It's a phospholribose that's there. Right? We've got a nucleotide at this point. We've got a purine nucleotide. And I will remind you later, you will see IMP again. IMP turns out to be very important in some transfer RNAs. IMP is an important nucleotide in some transfer RNAs. Well, to go from IMP to AMP and GMP is interesting and important. I'm going to go through it briefly, and I'll start next time with it also. But here's IMP. We see it's a branch point in going upwards to AMP or going downwards to GMP. Both can be made from IMP. Okay? Let's look what happens going up. If we start with IMP and we go over to AMP, GTP energy is necessary. If we start with IMP and we go over to GMP, we discover that ATP energy is necessary. Balance. If I have plenty of GTP, I'm going to go this way. If I have plenty of ATP, I'm going to go this way. Further, AMP right here will feedback inhibit this enzyme. I'm not giving you the name, but it will feedback inhibit this enzyme. AMP will feedback inhibit its own synthesis. GMP will inhibit its own synthesis by feedback inhibition. Okay? Yeah, I heard that sigh. All right? These are providing balance for us. Energy from GTP making AMP, energy from ATP making GMP, each one inhibiting its own synthesis. Now, I know this lecture is a bit of a nightmare, so I thought we would do a song at the end about nightmares. Okay? It's to the tune of an old Beatles song. Okay? It's an obscure Beatles song called Norwegian Wood. So please join me if you know it and sing it loud, because I don't sing very well. I answered 3B, but then I thought it might be C. Or was the false true? I can't undo. It makes me blue. It asked me to sing all the enzymes that regulate fat. As I wrote them down, I discovered I didn't know jack. I ought to give thanks, scoring some points, filling in blanks. I squirmed in my seat, feeling the heat, shuffling my feet. Professor then told me there wasn't a chance I would pass. So I started crying and fell through a big pane of glass. I suffered no harm, 'cause I awoke to my alarm. Oh, nothing compares to deadly scares, student nightmares. Okay. Happy weekend. Hey, Emily, how you doing? Did you know the song or not?
No.
No, I'm too old. I'm too old.
I like Beatles, but I don't know them.
Yep.